Hi, and welcome everyone. Before we begin today's meeting, there are a few housekeeping items we'd like to review. If you're not familiar with Zoom, all your meeting controls are in the lower menu bar. We ask that you please update your Zoom name to be your first and last name. You can change your name by opening the participant panel, select the person icon from the menu bar, find your name, which will appear at the top of the participant list, and when hovering over, select your name and more and rename. For attendees, you are not required to be on at webcam, and we ask that you keep your line muted for the duration of the meeting. Q&A will be facilitated by our session moderators. Please submit your questions or comments throughout the speaker's presentation in the chat window and select all. The moderators will ask your questions on your behalf, and we will try to get to as many as possible. If you require technical assistance, please feel free to contact me, the host, via the chat box, and I'd be happy to assist you. My name is Jennifer Schaefer, and I'd be happy to help you. I'd also like to note that today's webinar is being recorded and will be available online at a later date. And with that, I'd like to pass it over to Jessica, who will introduce our first speaker.
Hello. My name is Jessica Petrick, and I'm an assistant professor of medicine at the Boston University School of Medicine and Sloan Epidemiology Center. And myself, along with Dr. Neil Murphy, who's an epidemiologist in the Department of Epidemiology and Biostatistics at Imperial College London, will be the moderators for this session. And he'll introduce Dr. Pete Campbell before Dr. Campbell gives his talk. So now it is my pleasure to introduce Dr. Yin Cao, who is an associate professor in surgery in the Division of Public Health Sciences at the Washington University School of Medicine. Dr. Cao specializes in cancer epidemiology with the aim to uncover novel lifestyle, microbial, and genetic risk factors for cancer. The overarching goal of Dr. Cao's research is to develop precision-based cancer prevention and control strategies. Current projects in the Cao lab aim to incorporate high throughput technologies into large-scale collaborative epidemiology and clinical studies to address provocative questions in cancer prevention, including the etiology and early detection of early-onset colorectal cancers. Dr. Cao is currently the recipient of an R21 from NIAAA and a KO7 and R37 Merit Award from NCI. Please join me in welcoming Dr. Cao.
Thank you so much, Dr. Patrick, for your kind introduction. And also thanks to the organizing committee for inviting me here. It's such a great honor to speak in this seminar series, along with Dr. Campbell, Dr. Murphy, and Dr. Patrick. So today, so this is the outline for today. I'll briefly introduce the trends for early onset colorectal cancer and touch base on the current literature, including our findings in the etiology and prevention involving putative risk factors and emerging risk factors. I'll also briefly touch upon screening and early detection, as well as survivorship. So as we are familiar that the increasing incidence of early onset colorectal cancer was documented since early 1980. This is in sharp contrast to the decline in CRC incidence among age 65 and above, and not entirely driven by increased colonoscopy among younger adults, because mortality has also increased and colonoscopy utilization is very low in younger adults with only 13%. So in addition, a first cohort effect was documented. Elevated risk was observed among individuals born after 1950, and these first cohorts carry their elevated risk as they age. For instance, adults born in 1950 have twice the risk of colon cancer and 4 times the risk of rectal cancer compared with adults born around 1950. So as a consequence, the median age at CRC diagnosis has decreased from 72 years in early 2000 in the United States to 66 today. So strikingly similar increase in early onset CRC has also been observed in 18 other countries. And about 26% of early onset CRC were distant disease at diagnosis, compared to 20% for later onset CRC. And this is in part due to delayed diagnosis. However, studies, mostly hospital-based study also to reveal molecular differences between early versus later onset CRC. And at this point, there are no definitive conclusions, as you can see the slides here, but most of them showed a lower prevalence of BRAF mutation and higher frequency of TP53 among early onset colorectal cancer compared to later onset colorectal cancer. So from my understanding, there are three major knowledge gaps in early onset colorectal cancer. First, etiology and prevention. What are the risk factors and contributors to the rise of early onset colorectal cancer? Are there indeed molecular differences between early onset versus average or late onset CRC? The second challenge, more practical issue, what can we do in terms of screening and early detection? Given the potentially emerging molecular differences, is adenoma still the right target for early detection and screening? And although American Cancer Society and Preventive Survey Task Force recommended screening to start at age 45, but is that enough to mitigate the rising burden of early onset CRC? And what do we need to do for early detection? Finally, survivorship. For instance, how could we address disparities? And are there actionable molecular signature or signals unique to early onset CRC that merit precision-based therapeutical approaches? And also, very importantly, what can we do to meet the short-term as well as long-term needs of these younger patients? And in addition to these knowledge gaps, I think it's also important to acknowledge the unique challenges for early onset CRC research. Most importantly, the absolute risk and incidence for early onset CRC is very low. So it's difficult to study risk factors in a prospective fashion, especially as many existing large cohorts actually started recruit adults over age 50. So some emerging risk factors such as antibiotic use as well as environmental factors were not assessed historically. And today I'll introduce preliminary findings from our group as well as others using different data types and approaches. And the goal is really try to highlight many important areas that require collaboration. So let's start with risk factors and drivers. Given that colorectal cancer is a multifactorial disease, we expect the same for early onset colorectal cancer. So this table shows behavioral risk factors that have been identified for colorectal cancer, strengths of association, and temporal trends in the United States. So for risk factors to contribute to the rising incidence of early onset CRC, ideally, we would require parallel increase that preceded the rise of early onset CRC because it really takes time for the risk factors to play a role. And based on the temporal trends here and the directionality, we hypothesize that obesity, diabetes, and diet potentially as important putative risk factors that may contribute to the rising incidence of early onset CRC. So let's first take a look at obesity. The dramatic population level increase of obesity was actually not apparent until late 1970s, and this is about 10 to 15 years before the rise of early onset CRC. It's also important to note that if we look into by age cohort, then age 20 and 39 has the steepest and more than double increase in obesity prevalence, followed by age 2 to 19. And nowadays, the obesity prevalence for age 20 to 39 already reached the prevalence for age 40 to 59. So moving on, I'll present some of the findings from the Nurses' Health Study 2. Nurses' Health Study 2 collected rich data. It's a large prospective ongoing cohort of over 110,000 younger women, aged 25 to 42 in 1989, with updated information on lifestyle factors, medications, and diagnosis every 2 to 4 years. So in Nurses 2, we found that obese women had 93% increased risk of early onset colorectal cancer compared to women with normal weight. The relative risk for 5-unit increase in BMI is 1.2. And in contrast, when we look into CRC diagnosed after age 50, the association was much weaker, which was consistent with the estimate from World Cancer Research Fund and AICR report. So we also found both BMI at age 18 and the weight change since 18 were both associated with increased risk of early onset colorectal cancer. Excitingly, a recent EHR-based case control study in the Kaiser Permanente, Southern California, reported obesity was associated with increased risk of early onset CRC, with stronger association for colon cancer and rectal cancer, similar to our finding, but they have much larger sample size with around 1,000 early onset CRC. And our finding on BMI at age 18 were actually also in line with two prior studies among men in Israel and Sweden, showing that BMI in early adult life was associated with increased risk of colorectal cancer at younger ages. But at that time, they didn't look into CRC under age 50 exclusively. So, so the next question is, what are the critical time period or life stages? Studies are very limited at this point. Studies regarding birth weight are very conflict at this point. And in our analysis, we did not find any association between body shape at age 5 or 10 with risk of early onset colorectal cancer in nurses too. But we found that body shape at age 20 really matters. So we definitely need additional studies in looking into the time window. So along the line of metabolic dysregulation, my group also utilized claims data to examine metabolic syndrome and metabolic comorbid conditions that include hypertension, hyperlipidemia, hyperglycemia, and obesity. And we identified a linear relationship between the number of metabolic comorbid conditions and the risk of early onset CRC. Interestingly, this dose-response relationship is more apparent in early than later onset CRC. So we also look into diabetes and found a significant association between the two, especially for partially or completely uncontrolled or complicated diabetes. And this association was also largely driven by colon cancer. Interestingly, analysis from the Swedish national cohort also reports significant and somewhat stronger findings for diabetes and early onset CRC. And notably in Sweden, the risk of diabetes among younger adults is only about 1/4 of that in US younger adults. So along this line, we also examine sedentary TV watching time, which is a well-established risk factor for obesity and type 2 diabetes, but not a widely appreciated risk factor for colorectal cancer. TV watching in the US population has increased by almost 8 hours per week since late 1960s. So in nurses too, we found that sedentary TV watching time was associated with increased risk of early onset colorectal cancer. Women who spent more than 2 hours per day sitting watching TV had 69% increased risk of early onset CRC compared to women who spent less than 1 hour per day. So also notably, this positive association was more pronounced for rectal cancer compared to colon cancer. Now let's move a little bit to diet. We know that diet quality in the US population has steadily improved, but that was really driven by older adults. In younger adults, there are minimal improvement over time. Previous analysis from Dr. Andy Chan's group showed that Western diet was more associated with distal and rectal cancer and CRC, as well as CRC that are microsatellite stable. Based on these and based on the molecular features of early onset colorectal cancer, this leads us to hypothesize that diet quality may be more associated with early onset neoplasia. So again, in nurses too, we found adherence to a Western dietary pattern characterized by higher intake of processed meat, red meat butter, and high-fat dairy products, eggs, as well as refined grains, was associated with increased risk of any early onset adenoma, and this is driven by high-risk adenoma. Well, in contrast, prudent diet, which is rich in vegetables, fruits, whole grains, fish, and poultry, were associated with reduced risk of any early onset adenoma and high-risk adenoma. Interestingly, we further found consistent reduced risk across 3 recommendation-based dietary indexes, including the DASH diet, AMAD, and AHEI 2010. Taken together, we think these provide very preliminary evidence that unhealthy diet likely contribute to the rising incidence of early onset CRC. So in terms of modifiable putative risk factors associated with lower risk of early onset CRC, data in the VA population reported that aspirin use was associated with 33% reduced risk of early onset CRC. And analysis from the Gecko Consortium also showed similar findings for NSAID. In nurses too, we recently also reported that higher total vitamin D intake was associated with reduced risk of early onset CRC and adenoma. So now let's move on to emerging risk factors. I think at this point, there are multiple different hypotheses regarding emerging drivers. And this is a very nice review that basically showed that suggests that 4 metrics should be considered. The first one we have talked about, the exposure should increase in parallel and potentially before early onset CRC, before the rise of early onset CRC. Second, such trend is global because we saw 19 countries have increasing incidence. And third, there should be some molecular evidence, either animal or human, to show that this exposure have inflammatory or microbiome modifying properties, especially on the distal colon and rectum. And fourth, the exposure can occur anytime during the life course. And based on these, they hypothesized that antibiotics, high fructose corn syrup, synthetic dyes, MSG, titanium dioxide, these are potential possible candidates. However, epidemiology studies are very limited at this point. Regarding antibiotics, I think there are conflict findings so far, and which potentially in part due to the lack of assessment of cumulative antibiotic exposure throughout the life course. And we also tried a little bit on high fructose corn syrup for some preliminary findings. So this is basically showing that during the second half of the 20th century, average US per capita soft drink consumption basically increased by 5-fold. And when we look into age and birth cohort specific sugar-sweetened beverage consumption, it considerably increased for age under 40 from 1977 to 2001. And there are accumulating animal studies supporting the biological mechanism linking sugar-sweetened beverage and colorectal cancer. Specifically, SSB could initiate blood glucose response and insulin secretion, and potentially linked to insulin resistance, inflammation, obesity, and type 2 diabetes. And as I mentioned, these are potentially very important also for early onset colorectal cancer. And excitingly, recent animal studies also showed that high fructose corn syrup treated mice, they had substantial colon tumor growth with aggressive tumor grade. And interestingly, this is independent of obesity metabolic syndrome. And a recent paper in Nature also showed that this process is potentially mediated by expanded surface area of the gut, as well as high levels of blood lipids. So in nurses too, we found preliminary evidence showing that sugar-sweetened beverage is associated with increased risk of early onset CRC compared to individuals who consume less than 1 serving per week. Those who consume 2 or more servings per week had 2-fold increase in early onset CRC. We also found a preliminary signal that sugar-sweetened beverage intake during adolescence was associated with increased risk of early onset CRC. So just to put this into a broader context, that we actually also looked into replacement with other beverage. We found that replacement with artificial sweetened beverage, coffee, and reduced fat milk and total milk were potentially associated with lower risk of early onset CRC. So higher SSB intake were found to be associated with obesity throughout the life course, as well as diabetes and cardiovascular disease. although consumption in the US has leveled off actually after 2000. But if you look into the prevalence, over 30% of children and adolescents had more than 12 ounces per day, and young adults always had the highest average intake, and that's since early 2000. Disparity were also documented for Blacks and Blacks and Mexican Americans. So while taxes were found to be helpful to reduce SSB sales and consumption, currently only 70 cities are implementing SSB taxes. And in 2018, the US National Salt and Sugar Reduction Initiative proposed a government-supported voluntary national sugar reduction targets, which were recently, a couple of weeks ago, found to have a huge impact on lowering disease incidence as well as societal cost. So definitely we need a coordinated effort to further reduce SSB intake in addition to validation study of our findings. So moving on to microbiome, which is widely considered as a mediator for the rising incidence of early onset CRC. Thus far, I think direct evidence showing microbial differences between early onset CRC or later onset CRC at the tissue level, or using pre-diagnostic tools in a case control fashion are very limited. We definitely look forward to more research in this area. Previously, Dr. Andy Chan's team has developed sulfur microbial diet based on abundance of sulfur metabolizing bacteria in the house professional follow-up study. And these sulfur metabolizing bacteria are considered to be carcinogenic. And then we recently linked this sulfur microbial dietary pattern with risk of early onset adenoma and identified a 25% increase in the highest quartile compared to the lowest quartile. So moving on to the next steps, what can we do with emerging risk factors and drivers? We recently put up this review with Dr. Shuji Aquino and Kim Yang, potentially advocating a life course approach in addressing risk factors for early onset colorectal cancer. It's definitely important to initiate well-designed cohort or case control study with biospecimen collection. And I think as you can learn from Dr. Campbell today, harmonizing existing data as much as possible is also crucial for us to address the rising burden in early onset CRC. And at this point, there are many studies or leaders in the field looking into along this line to advance the field. And myself, as a younger generation epidemiologist, I'm lucky to be funded by the NCI Merit Award to look into life course epidemiology, proteomics markers, and precision screening strategies with the generous support from Dr. Andy Chan and the colon cancer team in the Nurses Health Study 2, and Dr. Zhang from the 1000 Community Cohort Study will utilize the rich data they have collected and to look into potential proteomics markers. Also, we'll work with the NCI CISNET colon cancer team with Dr. Ann Zauber and Dr. Iris Lansdorp-Wogeler. We'll look into how these risk factors potentially play a role in taking into account their temporality as well as their impact on the natural history of colon cancer to see how these risk factors potentially contribute and how much they contribute to the rising incidence of early onset CRC. And we will hear more from Dr. Campbell about his leadership and initiative within the NCI Cohort Consortium. And I would just want to highlight effort from Dr. Peters and Dr. Hayes in the GECKO Consortium. They have lead a couple of investigations that utilize the harmonized genetic as well as metadata from a large number of observational studies. So next question, are early onset colorectal cancer a different entity compared to later onset CRC? As mentioned earlier, prior studies were largely single institution based and limited in sample size. So they may have oversampled advanced diseases and included high-risk populations. So at this point, understanding the molecular landscape of early onset colorectal cancer through large, less selected population-based studies is still an unmet need. We are fortunate to work with the Gecko Consortium on this. We looked into the molecular feature of early onset colorectal cancer and difference according to age as CRC diagnosis, utilizing data from over 2,300 early onset CRC and over 10,000 later onset CRC with molecular tumor marker profiling. And these cases came from the Gecko Consortium and the Colon Cancer Family Registry. Most of them are population-based. We found that early onset CRC were more likely to be non-MSI high, SNP-negative, and BRAF wild-type tumors. And these analyses potentially support the continuum of features of early onset CRC according to age, rather than a sharp dichotomy at age 50. So next, we took a step further to see how we could use these molecular markers to infer pathway of progression in early onset CRC. This is a very important question because there are limited empirical evidence on the efficacy of CRC screening, especially among younger population. And the simulation in the used to support the ACS and task force guideline assumed all CRC or all early onset CRC arise from adenomas. So in data from Gecko and CCIFR, we classified tumor molecular signatures using just subtype, which is a well-accepted molecular framework for CRC progression. This particular subtype, just subtype 4, as shown here, is considered to reflect CRC arising from the traditional adenoma carcinoma sequence. As you can see here, the prevalence decreased with age, and these findings lend initial but strong support for the etiological relevance of early onset adenoma to early onset CRC. So now moving on to more of the implementation side. As we all know, this May, the task force for the first time recommended screening to initiate at age 45. And this is based on 3 independent macro simulation models that showed an efficient balance of life year gain and colorectal compared to the colorectal cancer colonoscopy burden. So, however, how to translate these recommendations into meaningful decrease in colorectal cancer incidence and mortality, especially among younger adults, can be a challenge and require a very coordinated effort. So the first challenge is that screening, as shown on the left side, screening is far from optimal, even for aged 50 to 59 at only 25%. So a recent study also looked into more than 2,000 early onset CRC. They found that 25% were eligible for early screening due to family history. However, only 2% were screened early. So what are the options to increase the yield of screening at younger ages? Led again by the Gecko Consortium, Dr. Su, Dr. Hayes, and Dr. Peters have showed that a polygenic risk score based on 95 GUS significant SNP was strongly associated with early onset colorectal cancer compared to colorectal cancer diagnosed at age 50 and above, as you see the green dots here. And we definitely look forward to more updates on the feasibility of such approach in real-world settings. So another issue that is that at least 50% of early onset CRC are actually diagnosed under age 45. So there's also an immediate need for early detection. At this point, we don't really know the set of red flag signs of symptoms for early onset CRC, although sparse studies suggest that they are more likely to have rectal bleeding and abdominal pain compared to later onset CRC. And we are not sure how these potentially early signs and symptoms, even a year before their diagnosis, can be used in combination to reduce the lead time for diagnosis. We don't know that yet. So finally, to survivorship, there are many studies looking into survival difference between early versus later onset CRC. Depending on the study design, most of them showed that early onset CRC actually have better or equal survival. And but importantly, even among patients with early stage disease, Black patients have worse survival compared to white patients, indicating the need for study differences in tumor biology or treatment response. So just to conclude, what we can do now, we definitely need more life course epidemiology studies or ex-post studies that measure everything we are exposed to. We also need genomics, microbiome, multi-omic studies. We also eagerly await for risk-based screening strategies. We also need to explore biological basis for disparity, sex difference, and age difference to potentially derive molecular-based therapeutics. And these initiatives are very exciting and definitely achievable with the effort from you all and support from the NCI and other funding agencies, but they all take time and need validations. In addition, I think we should definitely try to implement what we know in unprecedented speed because our younger generation or in the past seldom relate themselves to cancer, couldn't wait. We also note multiple increase in multiple other cancers among younger adults. So at the minimum, we need to help them reduce obesity, metabolic comorbid conditions, sedentary behavior, sugar-sweetened beverage use through policy, school and wellness program, and potentially community support. At the same time, we need to increase awareness among physicians and patients of the early signs of symptoms because 50% of the early onset CRC will not be detected even if we have perfect screening, which is not the case at this point. We also need to increase awareness potentially among the patients to let them know that potentially the signs of symptoms for early onset CRC to facilitate early detection. Finally, on the survival side, we need to address structural barriers in colorectal cancer screening and cancer care, improve genetic testing, and most importantly, we need physician support and provide support to our physicians so that they can improve cancer care and provide better support to meet the unique needs from the younger patients. So that's all what I have for today. And I would like to thank my mentors and collaborators who really paved the way for this research. They set up the infrastructure to allow younger generations to look into these provocative research questions. I would like to thank support, especially from the Nurses Health Study 2. Also, thanks to the support from NCI CISNET CRC team, Colorectal Consortium, CCFR, Southern Community Cohort Study, and funding support from the NCI and CCN, as well as Segment Cancer Center. Thank you.
Thank you so much, Dr. Kao, for the presentation. Just as a reminder to our attendees, please type your questions into the chat box and make sure that you're selecting everyone so that we receive your questions. And with that, I'll pass it over to Dr. Neil Murphy to introduce our next speaker.
So everyone, my name is Neil Murphy. I'm a scientist at the International Agency for Research on Cancer, and it's my pleasure to introduce the second presenter, and it will be given by Professor, the presentation will be given by Professor Peter Campbell. So Pete is currently a professor at Albert Einstein College of Medicine, and prior to this, he was a scientific director at the American Cancer Society, where he worked for over 12 years. He leads a program of research into GI cancers, obviously including colorectal cancer, using the CPS cohorts and other large-scale consortia resources. Pete, along with myself, is PI of the Colorectal Cancer Pooling Project, which I'm sure he's about to mention shortly. So Pete, thank you for presenting today, and the floor is yours.
Thanks very much, Neil. It's really a pleasure to be here. Thanks too to the other coordinators for this opportunity. And Yen, I have to say it was just an incredible amount of work that you and your group have done in such a really short period of time. And I'm going to pick up where Dr. Kao left off. In particular, I'm going to try to specifically address the title of this webinar and give sort of a 30,000-foot summary of where I think the field is now and where I hope that we go through some large collaborative projects. And next slide, please. So I want to start just sort of by laying the foundation. So first, I think that the trends of increased incidence mortality rates for colorectal cancer in adults under age 50, I think it's a very real phenomenon. I don't think this is a screening bias like we saw with prostate cancer and PSA testing in the 1990s or anything along those lines. Second, I think we've seen some really excellent work on etiology, but it's quite far from definitive. Dr. Cow's work here is probably the gold standard so far in this space, and those studies generally include about 150 or so cases. Sometimes studies have mixed adenomas with carcinomas. And we need to remember, I think, that it took enormous population level data sets to discover the descriptive trends that we're seeing. And I think we really need to invest sort of the same scale of resources to understand etiology. And further to that same point, I think identifying risk factors for early onset colorectal cancer should be a high priority, not the only priority, but a very high one. Of course, if we understand the causes of a disease, we're better positioned for primary and secondary prevention. And the fourth point here, sort of a continuation, I think, from the second point, but outside of the large descriptive data, I think as discipline, we need to be really cautious when we discuss findings from a few dozen early onset cases. It's generally pretty unlikely that a sampling frame of a dozen or few dozen highly selected patients can represent the thousands or even tens of thousands of patients that are diagnosed with this disease in any given year. And then the last sort of broad generalization that I want to make is that we know colorectal tumors are highly heterogeneous and multifactorial, and I think it's probably reasonable to start with the assumption that early onset sporadic colorectal cancer is too. Next slide, please. Now, thinking now about where I think the field is headed, or at least it could be headed with appropriate resources, first, I think we need to focus efforts on selecting the best study design for the intended purpose, the so-called fit for purpose expression. Sometimes a prospective cohort study is best, and sometimes it's even the only option to address a given research question. But there are other times where a case control study or a study based on electronic health records or medical records might be better. So many of our current cohort studies were launched in the 1990s, so we're missing important study data on potential risk factors, and I think you mentioned some of them, but they would include, early life exposure to antibiotics, maybe sleep patterns, exposure to maybe industrial detergents or chemicals. As well, are there any bona fide pre-diagnostic circulating blood markers for early onset colorectal cancer? Not to my knowledge, but that's definitely an area that we need to invest in as well. And if early onset colorectal tumors are heterogeneous, as Dr. Cow's work just showed us in that unpublished work, and also if we want to explore G by E interactions and so on, we're going to need really large sample size. Again, this is just a resource issue. We know how to do these studies. An enormous barrier that we need to address is the fact that most of this work in colorectal cancer etiology in general has been conducted in very white populations. So we need to include non-white participants in future studies and current studies, and determine if risk factors in those populations are similar, especially in populations where the disease burden is higher, such as in Black Americans, for example. And last, how do we implement these findings? And again, Dr. Cow ended her talk with some of those points. But can they be used for risk stratification to determine what people might benefit the most from screening? And if so, at what age? Or should we simply continue to recommend screening everybody at age 45 or 50 and miss a lot of very important disease burden that would occur in those even younger ages? Next slide, please. So thinking back to the point I made a minute or so ago with just a little bit more elaboration, first we know that colorectal cancer in primarily older adults has a lot of lifestyle behavioral risk factors. They're listed here on the right. So we see red meat, obesity, sedentary lifestyle, and so on. So it's a very multifactorial disease, but it's also a disease for which there are no major smoking gun risk factors. There's no risk factors of the magnitude that we see between, say, smoking and lung cancer. We also know that there's a strong inherited genetic component to this disease. So we see the obligatory Manhattan plot here on the left. We've identified somewhere between 150 to 200 germline genetic variants associated with colorectal cancer at a genome-wide level significance, and those are beyond the familial syndromes, of course. And last, we know that colorectal tumors are very heterogeneous on a molecular level, and we see that at the bottom just with the frequencies of various mutations in different tumor subtypes. And again, these findings are primarily from older adults, but I think this is important today because we really need a logical place to start when we think about the causes of early onset disease. Next slide, please. And Dr. Cal mentioned this paper, so I won't belabor it, but I think this is the study where Alexis Archambault from NYU and many others of us were interested in polygenic risk score, and we looked at associations with early versus late onset disease. And as Dr. Cal mentioned, this polygenic risk score was more strongly associated with early onset cancer than it was with later onset cancer. And for our purposes, there are probably at least two important implications here. One, I think this adds support to the hypothesis that at least the germline genetic predisposition to sporadic colorectal cancer is similar in younger and older disease. And then second, I think targeted GWASs, in younger cases and controls need to be done to see if there are any new loci for colorectal cancer in younger people beyond the known familial syndromes. Next slide, please. And here we're looking at the companion paper to the first. Same data set, slightly smaller sample size, but still something like 3,800 cases and 4,000 controls for the early onset analysis and tens of thousands of older cases and controls. And the goal here was to determine whether lifestyle and behavioral risk factors for late onset colorectal cancer were also associated with early onset disease. And if we scan top to bottom, perhaps most curiously, we see high BMI and smoking were not associated with early onset disease. But generally, most of the other risk factors showed the same pattern of association, although with perhaps a little bit less precision probably because of the smaller sample sizes. Next slide, please. And this is really intended as an example on why we need to pool resources and maybe also wait for some studies to mature a little bit. So please don't share or copy the slide, please. So these are results from CPS3 for the usual risk factors for colorectal cancer stratified by attained age. So CPS3 is a cohort study of 300,000 women and men in the US, conducted by my former institute, ACS. It ended enrollment in 2013, and here we're looking at cancer outcomes up to, I believe it's 2018. So we're looking at what I suspect might be a little bit of bias or maybe some chance for some of the under age 50 category. So that includes NSAID use being associated with a near doubling in risk of early onset colorectal cancer. That might likely be confounding by indication or reverse causation, meaning that people with GI pain or distress, they might be taking more NSAIDs as pain relievers. Similarly for alcohol, we see high consumption is associated with lower risk. I think that might be because people with early symptoms of bowel cancer might be avoiding alcohol consumption, so their rate is higher in the unexposed group. And again, we see that null association for BMI that gets stronger in the older two age groups. So I'm not sure if that's truly a null association that might persist with longer follow-up time, or if it's simply the result of reverse causation, perhaps from the shorter follow-up time. Next slide, please. And this is a study that, again, Dr. Cal mentioned briefly. This is from the VA Administrative Database. It included about 650 early onset cases and tens of thousands of controls. So this is a case control study design. We see increasing age and male sex are associated with higher risk. There's some suggestion that smoking is also associated with higher risk, but it's not quite statistically significant. And again, we see aspirin intake is associated with lower risk. We saw that in the GECKO study. And BMI here goes actually in the opposite direction to what we see in older onset disease and what we saw in many of the studies presented by Dr. Cow. So, and then here in post-hoc analyses on the right, they found that weight loss around the time of colonoscopy was associated with higher risk. So here, this inverse association in the middle might be driven really by reverse causation from illness-associated weight loss. And the takeaway here, I think, is that timing of weight measurement relative to diagnosis really matters. Next slide, please. And just to make one final point on BMI and early onset disease, here we see a meta-analysis published this year by our colleagues in Germany. It included only 4 studies in this graphic, and none of the studies that I just mentioned were included because they were too new. But here we conclude that higher BMI was associated with higher risks of early onset colorectal cancer, in contrast to the null and even inverse association from the last 2 published studies. And I only mention this conflict to really underscore the fact that we really are in the early days of understanding the risk factors for early onset colorectal cancer, and we need to do bigger, more definitive studies. And if you remember the early days of candidate gene or candidate SNP studies from the early to mid 2000s, I really feel like we're repeating history to some extent with some of these smaller studies here. And next study, please. There are probably many ways to do bigger and more impactful research in this area. And maybe one of the most efficient approaches is to combine multiple prospective cohort studies together using the framework created by the NCI Cohort Consortium. So that's what we started about 1.5 years or well, maybe 2 years ago now, I think. And our goal is stated here, but it is essentially an international resource for understanding the etiology of colorectal cancer according to age at onset. So we're interested in questionnaire-based variables, especially those that have become more common in recent decades, that Dr. Cow pointed out. And we're also interested in biomarker discovery. And in addition to Neil and Yin, who are on this webinar, I'd like to acknowledge our colleagues, Andy Chan, Mark Gunter, Richard Hayes, and Ricky Peters for their insights and leadership in helping us create this, this resource. Next slide, please. I suspect given this audience, I probably don't need to convince you of our justification here, but these 3 points basically distill into the fact that early onset disease is still quite rare and few cohorts have sufficient numbers to do meaningful analyses on their own. Next slide, please. I apologize for the font size here, but the important summary points are that we have 26 cohorts participating. I think 25 of them are listed here. We have a total of nearly 3.5 million study participants from, a reasonable amount of the world, not much more than a reasonable amount, I guess. And perhaps most importantly, this includes just over 2,600 early onset colorectal cancer cases, 15,000 middle onset cases, so that's ages 50 to 64 as we've defined it, and then a bit over 25,000 older onset cases. So those are cases age 65 and older. Next slide, please. And I don't want to go too deeply into some of the rabbit holes in our specific aims for grant submissions, but broadly, we're interested in prospectively identifying risk factors for early onset compared to later onset colorectal cancer. And these efforts will include estimating relative risks and absolute risks, as well as doing some risk prediction modeling. And many of the cohorts will also have biospecimens, and we hope to leverage those for future studies. Next slide, please. And I know many of us are firmly entrenched in the cohort world, but I think this is a research area that could probably benefit from really well-designed and executed case control studies as well, really to be complementary of the work that we're doing in the cohorts. So some of those complementary areas or strengths of case control studies might include the fact that you can design questionnaires to capture some of the more detailed hypotheses on some of the potential causes for early onset colorectal cancer that aren't typically captured by current cohorts. So that would certainly include, early life antibiotic use and some of the other earlier life exposures that have been mentioned. You could get more detailed timely data on family history of cancer, which could help you parse out familial from sporadic early onset disease. The data harmonization processes wouldn't be simply restricted to, the most common denominator. For smoking, for example, you can get much more detailed information on duration, intensity, age at initiation. In terms of survival and prognosis studies, surgical treatment data could probably be collected in detail through electronic health records for most for most areas that we might be interested in. Could definitely collect much richer and uniformly collected biospecimens. And last, and maybe most importantly, we could collect information from non-white study participants, which we absolutely simply need to do. And is indeed probably one of the larger motivating factors for me moving to Einstein recently. And next slide, please. And with that, I just wanted to say thank you for your attention, and we are interested in hearing your questions.
Thanks, Pete, for your presentation and introducing the Colorectal Cancer Pooling Project. It's really lovely. Let me just put my video on, apologies. So we have time for a Q&A session led by myself and Jessica. So thank you for those who have submitted questions. So I'll kick things off. So we had a question for you, Pete, regarding the CPS3 analysis that you presented. It says, for potential reverse causation, did you exclude colorectal cancer cases diagnosed a few years after baseline? And how might that have changed the findings, especially for alcohol?
The short question is no, we did not, because most of those cases occurred shortly after baseline. So if we excluded them, we would have almost no one left. So we had, just as a reminder, we ended follow-up in 2013. Most of those 300,000 people were enrolled in 2011 to 2013. We only had follow-up through to 2018. So at max, or on average, we have about 5, maybe 6 years of follow-up. If we excluded participants diagnosed within 2-3 years of follow-up, we would lose sample size entirely, but we would probably see those inverse or unexpected associations go away. Generally, that's what one sees.
So incidence is continuing for early onset colorectal cancer is continuing to rise in individuals less than 45. And as Dr. Cow noted, that about 50% of early onset colorectal cancers are occurring in individuals less than 45 years of age. So do you think that we're headed towards eventually continuing to recommend like even younger ages of screening? Or do you think we're headed towards some sort of like a targeted risk stratification for screening of less than 45-year-olds?
Who's that one for? Is that for Ian? Do you want to take that one?
Oh yeah, sure. That's definitely a great question. I think both are possibilities, but I do see that potentially targeted screening trying to identify individuals with higher risk might be a more feasible approach, especially given that we have known, as I have shown, Dr. Peters has led this really large study using polygenic risk score. I think we are really in a relatively mature stage to move that to a more implementation stage. And I think on the higher level question in terms of future policy, I think it really depends on the net balance in terms of moving the target lower and how much life years that will help save and compared to the colonoscopy burden, as I mentioned. But I would imagine moving to age 40 would at least take another 5 to 10 years while we have more evidence in the younger age group. At the same time, as an emphasize, there should be more efforts in this age range, younger than age 45, trying to identify their cluster of symptoms and early signs, and in what way we can help the early detection with these patients. Pete, do you have anything to add on?
Yeah, I mean, there's, just two parts. One, and more briefly, I think we need to work smarter, not harder. I don't think lowering the recommendation to age 40 is going to happen anytime soon. If you refer to a paper, I believe it's published in Gastro in 2019 by Rocky Schoen, was the senior author. I believe one of his medical students was the first author, but he did sort of an economics analysis on, what is the cost benefit ratio of moving the age at which we start screening to earlier and earlier intervals. And as outcomes become rarer and rarer, and the potential side effects like bowel perforation, cost, and capacity, which are all very real limitations. When you start weighing those in, it gets really tough to justify doing population-wide screening in asymptomatic people below many ages. And I won't put a number on that age because I think it's a little bit subjective what age you pick.
Okay, I think we've got time for maybe for one more question. So the birth cohort effect, which you both mentioned and touched upon, suggests that early life or cumulative life exposures may be driving the higher rates of disease in young adults. And I'm wondering to have your opinion or perspectives on how best we can study early life-caused exposures better. Obviously, it's a real challenge.
So can I let Ian answer that? But first, can I say that this might also be a period effect, but screening has blunted the period effect in people over age 50. It's not a popular opinion, but it does fit the data.
Yeah, I agree with Pete. I think in terms of studying early life exposures, for the things that are captured in medical record, I think there's definitely a need to push those studies, especially states with registry and EHR data combined, and to look into birth weight and all those aspects, and even perinatal exposures. On the other hand, I think as Pete mentioned in our C2P2 initiative that he's leading and Neil is leading, we are potentially also looking into or trying to harmonize early life exposures. But technically, it's a very challenging question using existing data. There's highly likely that what we captured using these existing data probably doesn't really matter, or they serve as confounders. So we definitely need smart design of new studies in terms of asking people to recall new exposures, or using historical data like the environmental data to try to put those things up from using more innovative approach.
I'm going to take the prerogative to ask this one last question that came in. The popular media has been highlighting a lot of early onset colorectal cancer cases in very healthy, non-obese individuals. So can you guys just summarize what you think are the leading non-BMI related factors for early onset colorectal cancer, and then we'll wrap up.
Oh, that's a hard one. I don't think the data support it. I think, in the 3 studies that I showed, BMI doesn't look like a terribly convincing risk factor. I think if you consider the descriptive epidemiology, it's primarily it's rectal cancer that is really increasing at the steepest rate. BMI and rectal cancer have a really weak association. So I think if we rely on the descriptive data alone, maybe we should have expected BMI not to be the hugest culprit, although we haven't ruled it out. I like antibiotic use, and if I had to put my money on one, it would be changes to the gut microbiome that have occurred because of a multitude of insults from age 0, or maybe even in utero, to age 50. So I'm going to hedge my bets on a lot of stuff.
Yeah, I totally agree. I think we don't have definitive conclusions, and even for BMI, but I think based on I presented the existing literature out there, including ours, I think the key message is that it's never too late to intervene on what we know, like the obesity, sedentary metabolic syndrome, increasing risk of diabetes. It may not be very specific to a broader extent to early onset colorectal cancer, maybe related to even other rising GI cancers. But the point is that we are really in a stage that while we explore the new potential emerging causes, we also need to mitigate on these issues that definitely a problem for our younger cohorts, especially those born after 1970 or 80. So I think there's definitely a need, especially for epidemiologists, to push that to practice as well. So I would like to thank everybody who joined us as well. I think with so many audience online, we definitely have a lot of avenue to implement what we know, actually what we contribute to the field, and to really change the picture for the younger generation.
Well, thank you so much to Drs. Cow and Campbell for the presentations, and thank you to all the attendees. And there'll be some final announcements here, but we hope to see you at the next webinar, which is scheduled for October 20th, examining measurements of aging and to use in cancer cohort studies.
Yes, thank you everyone for joining Today's session was recorded and will be posted online at a later date. We'll make you aware of once that's available. If you have any new topic suggestions, please feel free to email them to the inbox that appears on your screen. And with that, I'd like to thank our speakers and our moderators again today for a great discussion. Thank you, everybody.